I hope you're all well. I'm Mirta, Secretary of the BioSoc. Thank you very much for joining us for this talk. We would like to thank our first corporate sponsor of the year, CRISPR Biotech Engineering, an early stage. I hope you're all well. I'm Mirta, Secretary of the BioSoc. Thank you very much. Genome editing company using CRISPR-Cas9 to develop immunogenic-based therapies, as well as providing educational resources. Today, we are honored to have Dr. Philipp Engel as our first speaker. He's an associate professor at Lausanne University and a leading microbiome researcher focusing on social bees as a model for understanding the evolution and function of microbial communities. His talk today will give an overview on bee microbiome research and summarize recent projects his group have been carrying out. The talk will be followed by a Q&A session. If you're on YouTube, feel free to post your questions throughout the talk. If you're on Zoom, you could ask your questions in person, post them on the chat, or send them privately to me. Without further ado, let's welcome Dr. Engel to deliver his talk.
Hey there, thanks a lot, Mirta, for this nice introduction, and thanks a lot for inviting me. My talk will be about bees and their gut symbionts. Our lab works actually at the interface of computational biology and wet lab experiments. And I guess most of you know that microbiomes or microbial communities, which you find in nature, are super important for ecosystem functions. They're important for our health, for example, in our human gut. However, one thing they have most of these communities have in common, at least the ones in the mammalian gut, is that they're extremely complex. So it's very challenging to study natural microbiomes which are out there. And when we take the mammalian or the human gut as an example, we have noted that there's an extremely high diversity. So each of us has about 100 to 500 different species in the gut. That by itself is not necessarily a problem, but the problem is, or the challenge is, that each of us has a different set. So there's a high variability, and each of us shares actually only a fraction of the microbiome. And this makes it very difficult, to find out how these communities are working in one case and how we can translate that knowledge to another case. Another challenge is that many of these microbes are not culturable. To study host-associated microbiomes, you need to generate microbiota-free animals, so animals which are lacking these bacteria or these microbes, which you can then use to inoculate with a specific set of microbes to study their function. So this has been feasible for a couple of model systems, but there are many other interesting animals out there for which this is not possible yet. And another important aspect is that the phenotypes of these microbial communities are not always obvious. So they're facultative symbionts, which means the animal can actually live without these microbes. At least we can, for example, raise germ-free mice, and they're surviving, right? So the phenotypes are not obvious, and the symbionts and their functions are redundant. So many different symbionts can do the same thing. And this makes it very hard to figure out what the microbiota is doing and what every member in these communities is doing. Another thing is that this community is not exclusively composed of beneficial microbes, so microbes from which we benefit, which would be here on the right side towards the mutualism. But there are probably also a couple of nasty bacteria in our gut microbiota, so pathogenic microbes, and some may be commensal. The fact is that there's a thin line of what a microbe is and how it interacts with the mammalian host, and there's a continuum from being virulent towards being beneficial. And this interaction may also change depending on the context, depending on your diet, depending on which other microbes are present, depending on the condition of the host, whether microbe is beneficial or maybe even detrimental for you. So the bottom line of this slide is that we need simple and especially tractable model systems to study microbiomes, especially gut microbiomes. And many researchers all over the world are using, for example, germ-free mice, and they associate these mice with a defined set of species, be it isolates from human beings or isolates from the mouse gut. So these are relatively, let's say, artificial situations, because you're not reconstituting the natural microbiota, but you're reconstituting a artificial community from a few selected strains. So for this reason, we are working on the gut microbiome of social bees, specifically the honeybees. And when I say honeybee, I mean Apis mellifera, which is the Western honeybee. Over the last few years, it has been found out that honeybees actually harbor a relatively simple gut microbial community, which is composed of, let's say, 8 to 10 phylotypes. So phylotypes are bacterial groups which share more than about 97% sequence identity in their 16S rRNA gene. So the 16S rRNA gene is universal marker gene, which you can use to describe taxonomic diversity in microbiomes. We will come back to that in a minute. So these 8 to 10 different phylotypes are shown here. So they're called Guillemella or Freshella or Snodgrassella or Lactobacillus from 5 or Bartonella apis. So most of these bacteria are specifically found in honeybees or social bees in general. They have not been found in any other environments yet. And over the last few years, researchers, including our group, have given them their own names, because, as I said before, these lineages have not been found before. So they're highly specialized microbes, which so far have only been found in social bees. So this slide is about like introducing you to the honeybee life cycle and the associated microbiome. I guess most of you know a lot already about honeybees. So a honeybee colony consists of thousands of different individuals. Typically, you have a single queen, you have a couple of drones, which are adult male bees. But the majority of a honeybee colony consists of adult worker bees, female worker bees. And so the life cycle starts with the queen laying an egg into these brood cells. And then a bee larva develops over time, pupation starts, and after about 21 days, a new adult bee, female worker bee, will emerge, okay? And people have looked at the gut microbiome of these larval stages over time, and the bottom line is that the microbiota of these larvae is relatively erratic. So you sometimes find this species, another time you find another species, and although the total bacterial biomass is relatively low. What happens is when bees pupate, they shed most of their gut lining, and thereby they also shed this microbiota. So at the pupal stage, we believe that these bees are actually void of gut bacteria. And so is the adult bee when she emerges, okay? And then what people have observed is that in the first few days of the adult life stage of these worker bees, they pick up a lot of bacterial biomass, and the gut gets heavily colonized by these different microbiota members I've shown you before. And we believe this is mainly driven by social interactions between different bee, oral trophallaxis, but also just exposure to the hive environment, as well as fecal oral road with older nest mates. Okay, so you see that the social context of a bee colony apparently or probably plays an important role in the acquisition and in the transmission of this microbiota from one generation to the other. So then when an adult bee has emerged, she typically stays in the hive. We call these the nurse bees. They take care of the brood. They feed the brood, they feed the queen, and then they take over tasks such as guarding. So they guard the hive. And the last task they're fulfilling is foraging. So the most risky task they do at the end of their life stage. So about 2 to 3 or 4 weeks, depending on the stage of the colony. These worker bees will transform into foragers and go out and forage for pollen and nectar. So at least at the phylotype level, the microbiota, once it's established here at about 4 to 5 days, does not change a lot anymore over the course of the life stage. And this is shown with the next slide here. So what you see here is the gut microbiota composition of adult female worker bees, foragers, nurses, and we also included here winter bees. So winter bees are bees in the winter, which stay in the hive and build this kind of winter cluster to keep warm. And although you see, you know, that you probably can appreciate that there are some shifts in relative abundance of these different, of these 8 to 10 different community members here, the overall pattern remains the same. So most of the community is really dominated by these few bacterial phylotypes. And these were, I guess, 14 different colonies sampled in Western Switzerland, but these kind of analyses have been done all over the place and the world, and we always find the same community members. So in contrast to the human gut microbiome, we have here a relatively stable gut microbiome. Wherever you sample these bees, you will find the same bacterial phylotypes. So these bacteria do colonize specific niches in the gut. So what you see here is a dissected bee gut with a foregut, the so-called crop, the midgut, which is then followed starting here by the hindgut, which is, you know, split into the pylorus, ileum, and the rectum. And certain bacteria are predominantly found in the rectum. Others are actually found in the ileum, which is this part here. So this is a fish microscopy image where we use fluorescent probes to label 2 specific members. One is Guillemella, the other one is Snodgrassella. And I'm not sure if you can see it very well, but what you see here is actually the gut tissue. So the gut takes a structure of a star-like structure, and you have these evaginations here. So this is the gut lumen, and you have these evaginations here. And what you see here, this thick layer, are actually bacteria colonizing on top of the host epithelium as a very thick bacterial layer, consisting in this part of the gut mainly of these 2 bacterial species. So here we have, you know, a batch of Snodgrassella colonizing the host epithelium. Here, Guillemella, with some Snodgrassella in between. So what this tells us by just looking at this is that apparently these bacteria are really interacting intimately with the host and also among each other, which suggests, you know, that they're really engaging in some kind of symbiosis. And many different papers over the last few years have accumulated evidence that the gut microbiota of bees, as in many other animals, plays an important role for bee biology and bee health. So, you know, using different types of experiments, researchers have shown that the microbiota impacts body or gut weight gain of adult bees. There are differences in simple behaviors, such as response to sugar water and even water. And people have shown that the insulin-like pathway is affected by the microbiota. It was also shown that bees with a normal microbiota have an increased colonization resistance, which means they're more resistant to invasion by pathogens, by bacterial pathogens. People have shown that the immune system is affected by the gut microbiota, and that also certain metabolite levels in the bloodstream and the hemolymph of bees is affected by the gut microbiota. So we have some good evidence that the microbiota is really impacting bee health. On the other side, we have very well understood what happens in the gut. So as I told you before, adult bees, they actually eat mostly pollen and nectar or honey. So nectar is stored in the colony. And the bacteria in the gut, they extract nutrients from these dietary resources, mostly or to big part sugars and sugar acids or complex polysaccharides. And they ferment them to large part into organic acids, short-chain fatty acids, but also other organic acids. And we know from the human gut that short-chain fatty acids is one of the most important bacterial products which affect mammalian or human health. And we believe that this is not different in the bee gut. However, we currently have not really understood how, you know, the metabolic functions or what is happening in the gut at the level of the microbiota is actually leading to this beneficial impact on the host. So we are pretty sure that what's going on in the gut has an impact on the host, but we don't know by which mechanism this is really mediated. What we know from a couple of really nice studies is that when you disturb the microbiota, you see actually that the bees start to become problems, especially in terms of colonization resistance to pathogens. So this has been shown with, for example, agrochemicals, pesticides, antibiotics. When you expose the bees to these kind of chemicals, you disturb the microbiota in its composition and also in its total abundance, and thereby you change the beneficial effects of the microbiota on the hosts. So one really nice thing about bees is not only, you know, that they're important for ecosystem and because of their pollination service, but also because their microbiota is really experimentally tractable, and this is really nice. So on one side, we can generate so-called microbiota depleted, or some people call it microbiota free bees, and we do this in the laboratory by removing bees from the hive when they're at this pupal stage. I told you before, this pupal stage, they don't have any gut, they don't have any gut bacteria. And when we remove these pupae from the hive and let them emerge in the laboratory, they will be born, they will become adults without gut microbiota, okay? On the other side, we were very lucky with the bee system because we can culture most of these bacteria and these phenotypes in the laboratory, and our lab and many other labs over the world have now most of these species in strain collections in the -80 freezers. So what we can do is we can grow them up on these agar plates, and we can inoculate our microbiota depleted bees with a single strain or a combination of different strains, and then do experiments and understand how these bacteria causally, for example, affect certain metabolic activities in the gut or bee health. So this is how it looks like in our laboratory. So here you see such a frame with brood cells, which we have taken from our honeybee colonies, which we have just outside of the campus. And then you open these brood cells with forceps or little tweezers, and then you carefully pull out these pupae. You need to pick the right pupal stage, and you put them into these Tupperware boxes, modified Tupperware boxes, and you put them into an incubator and let them emerge. So it takes about 2 to 3 days until these bees have emerged and have reached adulthood and become adult bees. We then put these emerged bees into these so-called cup cages. We feed them sugar water. You can't see this very well, but here's a little tube which contains sugar water. And we also put a trough which contains sterilized pollen, so that the bees also have a pollen diet. And then you can do experiments. So you can take these newly emerged bees and feed them with a pipetter a defined number of bacterial cells or a combination of different bacterial cells, and then see how they impact the host phenotype. Important is that we are obviously controlling the status of our microbiota depleted bees. So we are always plating a couple of these bees to make sure that there's no contamination. So in this bee, for example, apparently something has either survived the pupal stage, the metamorphosis, or has come in as a contamination from the laboratory. So cup cage like this, we would obviously exclude from our experiments. So with this kind of setup, we can do experiments as the following one. So this is just one example. So in this case, we wanted to know, what kind of gut metabolites are influenced by the gut microbiota, and which of the different gut microbiota members contributes which metabolite in the gut. So the experimental setup was as follows. So we generated our microbiota depleted newly emerged bees. We then either kept them without bacteria, so we left them microbiota depleted, or we colonized them with a reconstituted microbiota, so with a defined set of strains we have in the freezer of all the different species of the community, or we associated them with individual strains, so with individual species of the major community members. We call these the monocolonizations, okay? So each bee is only colonized by one species at a time. And then we waited 10 days, the microbiota establishes, and we feed them a pollen diet. And then after 10 days, we dissected their guts and we subjected these guts to metabolite analysis, untargeted metabolomics, where you can detect thousands of different ions, which you can then assign to different gut metabolites. And the questions here were the following ones. So you can compare the metabolite profile in the gut of colonized bees versus these microbiota depleted bees to ask the question, you know, what kind of metabolites are produced by the microbiota and which ones are consumed generally. And then you can also go to the monocolonizations, ask the question, okay, which of the different community members is actually responsible for which metabolic activity? So this is published research from our laboratory. And obviously, this is an omics study with a lot of results and a lot of metabolites you can identify and a lot of metabolite changes. This is kind of an overall summary. So we identified a lot of pollen-derived substrates these bacteria are utilizing. And we also identified a number of interesting microbial products which are accumulating in the gut when the microbiota is present. For example, aromatic compounds, short-chain fatty acids. On the side of the substrates, we identified that flavonoids are, for example, converted by the microbiota. And since we not only did microbiota depleted bees versus the whole community, but also these monocolonizations, we were also able to explain which of the community members, so who is doing what, who of the community member is actually responsible for which metabolic change. So we are currently looking into which of these metabolites are actually taken up by the host and which of these metabolites have an impact on the host health. And the other way around, you can also ask the question whether the host actually contributes certain nutrients to the gut microbiota and feeds the microbiota certain metabolites. So one important thing I want to say, so the question is, what can we learn from all this, right? On one side, we can learn obviously things about bee health, but my lab especially is interested in fundamental questions also about microbiota research. And one important thing I want to tell you here is that although the human gut microbiota is composed of different species and phylotypes than what we find in bees, there are actually a couple of common features. So we actually believe what we learn from a bee system, we can actually translate to other animals, including humans. So for example, in humans and in bees, we have a highly specialized microbiota. These bacteria mostly cannot be found in other environments. Short-chain fatty acids are produced in both systems. The gut is anaerobe in both systems. And there's, for example, also clear evidence for interaction with the host immune system. So there are a lot of shared features. On the other hand, as I told you before, the honeybee gut microbiota is much more simple. Most of these bacteria are culturable, and we can also do experiments with the host. So these are aspects which are challenging, obviously, to study in humans, where you have hundreds of different species which are coexisting. So with this kind of general introduction, I hope I could give you, an idea why it is interesting and relevant to study bee microbiomes. On one side, this is because it helps us to get a general understanding of how microbiomes functions. So we can understand, what is the microbiota doing, but also which member of the microbiota is doing what. And on the other hand, also I haven't talked about much, but with what kind of interactions are actually taking place within the microbiota. And the main thing is that this system is experimentally tractable and relatively simple compared to other gut microbial communities. On the other hand, the bee microbiota research can really contribute to improving bee health. And this is not the main topic of our lab, but there has been some very, very important publication this year by the lab of Nancy Moran, and I really recommend you to look at these papers, where they have shown that you can actually apply synthetic biology to bee gut microbiota members, genetically modify them in a way that they increase the resistance of bees towards two very important pathogen or parasites of honeybees, which is the deformed wing virus and the parasitic mite, the varroa mite, which transmits this virus between bees. And these are mites, and these viruses are really almost the #1 enemies of honeybees nowadays. And we see a lot of colony losses because of these viruses and mites. And we really need to treat the colonies against these pathogens so that they actually survive the winter. So although these are genetically modified organisms by which this increased resistance is mediated, this is a super interesting approach for the future, which gives hope that we can actually improve bee health with such molecular tools. So with this, I would like to switch gears a little bit and talk more about one specific aspect my lab is interested in, and for which we are looking at the honeybee gut microbiota and at social bees in general. And this is about diversity in microbiomes. So we always say, you know, microbial communities are everywhere. You can find them in every environment, and they're generally extremely complex and diverse. This is true, but we have also now learned from honeybees that in honeybees, it looks like the microbiota is, for example, less complex and less diverse than in the human gut. And the question is, a very general question is, what determines actually diversity in microbiomes? This starts the question, how do we actually quantify and measure diversity? I would argue that we are still not very good in measuring diversity in microbiomes, leading then to the question, how does diversity emerge in terms of evolution? How can it be maintained? How can so many different bacterial species and strains actually coexist in a given environment? So what are the driving factors? So these are very general questions which we are looking at by looking at social bees. So why do I say, you know, it's actually difficult to measure diversity in microbiomes? Well, this is because we have mostly, in the last few years, carried out so-called 16S rRNA sequence analysis. And I would argue that with these kind of analysis, you get a very good overview of diversity across many, many different samples and many different microbiomes, but you're actually only looking at the tip of the iceberg. So that would be the entire diversity which is out there, but with 16S rRNA analysis, you can only look at the tip of that iceberg, and there's a lot of diversity hidden underneath. So how are 16S rRNA sequence analysis working? So the idea is that you take samples from different microbiomes, you extract genomic DNA, and what you do then is you use universal primers, which are primers which are supposed to amplify the 16S rRNA gene of any bacterial species, and you use these primers to amplify up a little fragment, a short fragment of the 16S rRNA gene of your sample. And if these primers are near to all bacteria in your sample, you will have a PCR product which represents the whole diversity in the sample. And you sequence that PCR product with Illumina sequencing technology, and then you cluster these Illumina sequence reads of the 16S rRNA gene based on similarity, and you cluster them into operational taxonomic units, or you define amplicon sequence variants. And with the help of this marker gene and the relative abundance of different variants in your sample, you can then determine what is the taxonomic composition of your microbiome. However, we know from literature and from genomic analysis that within a given species or a genus, you can have many different strains. And these strains carry different genes in their genome. There's a lot of genomic diversity out there. And if you want to understand, what microbiomes are doing in nature and to understand how they have evolved, you need to look at the functional gene content, because this is where evolution acts on. This is what determines the function of a microbiome in a given habitat. So you really need to look beyond the 16S rRNA level to understand how much diversity is there and what this diversity may be doing in a given ecosystem. So you can do that by doing shotgun metagenomics. So here again, you have your samples from different microbiomes, you extract genomic DNA, but instead of amplifying just the 16S rRNA gene, you sequence everything which is there. And if you sequence everything, you get all the genetic information sequenced, you get information about every different gene in every different strain, and you actually get a picture of the entire taxonomic diversity which is in your sample. The drawback of this method obviously is that as more complex your microbiome is, as deeper you need to sequence, because now you not only sequence one little gene, but you sequence the entire genome of a given species. So the sequencing effort is considerably larger. However, since sequencing costs have been going down a lot over the last few years, this is really now feasible for a lot of different samples. Another challenge associated with shotgun metagenomics is the analysis. So it's not straightforward to quantitatively analyze shotgun metagenomic data. You can obviously appreciate if you sequence everything, you will have a huge diversity of sequence information in your sample, and it's very challenging to make sense out of this. So why do I tell you all this? Because before I just told you that, well, the honeybee gut microbiota is simple. You have 8 to 10 different phylotypes. However, very early on, when I started to do research on the honeybee gut microbiota, we realized that actually there's a lot of strain level diversity in each of these different phylotypes. But at that point of our research, we really didn't understand how this strain level diversity is structured and how it's distributed in nature in these different bee populations. So what do I mean with structure of the strain level diversity or diversity in general? So this is a schematic kind of phylogenetic tree. So when you do 16S rRNA analysis and cluster these reads on the phylotype level, you would maybe identify 2 different lineages in your sample, so 2 different community members. So a given community member can consist of just a few closely related strains, right? However, another phylotype may consist of 2 or several deep branching lineages, sub-lineages, and they have they've had enough time during evolution to actually irreversibly separate from each other, which is indicated here by these long branches, so that they represent sequence discrete populations. So populations you can discriminate from each other based on their sequence information. So there are no strains here sitting in between, and you can clearly say, well, these strains here belong to this lineage and these strains here belong to that lineage. Obviously, when you're doing 16S rRNA analysis, you will just see 2 community members. But in reality, you have different sublineages, or SDP, sequence discrete populations, or you can also call them species. And in each species, you can have several strains which can have different genes in their genome, which are functionally relevant. So this is the structure of genomic diversity, or strain level diversity. And now you can also then ask the question, once you know how much diversity is there, how is it distributed in the environment? So for example, every bee, so a given bee you are sampling may have all the strains of all the different sublineages. So from this, you would conclude, well, they can all coexist in the same habitat. Maybe they have different niches, maybe they have different functions. So they seem not to compete with each other for the same things, and therefore they can coexist. If you're looking at this scenario here, B2, you actually see that you only have 1 strain and only 1 strain per sublineage, okay? So if you would find a pattern like this, well, then you would say it looks like in a given bee, you can only have 1 strain per sublineage coexist. And then there can be many different other patterns. What I want to say with this, once you know how your diversity is structured and how it's distributed in the environment, you can start to form hypotheses about how this diversity may have evolved and how this diversity is maintained in the ecosystem. And to do so, we obviously want to do that using shotgun metagenomics data. So why is it so interesting to look at this in social bees? So before I told you, the first reason is that the phylotypes seem to consist of many different strains. The other aspect is that honeybees live in these large colonies of thousands of different bees. And every little bee, can be seen as a little island which carries a certain set of bacteria. So since bees of the same colony supposedly have much more social interactions among each other than bees from another colony, you could argue, or you could believe, that, for example, in a given colony has a kind of colony-specific strain composition and signature, okay? So all the bees from a given colony look the same. So you have the green colony and the yellow colony. However, it is also possible that every bee carries a different set of strains, and that some bees, although they are sitting in different colonies, are more similar to each other in terms of the microbiota than they are to their nest mates, okay? So we would have individualized strain profiles, and each of these different profiles is present in a given colony. Obviously, this can have functional implications, because if this colony harbors more beneficial strains, then maybe that colony is much more healthier than this colony, and that would be important to know for a beekeeper. On the other hand, if you find a pattern like this, then maybe some bees in your colony are more healthy than others, so it would be better to have more of this type, for example. So how did we approach this? So this is work which was really spearheaded by a postdoc in the lab, Kirsten Ellegard, and she developed kind of a pipeline which allowed her to enrich bacterial DNA from individual honeybees, and then shotgun sequence this enriched DNA on one hand. And the other part of her approach was that we collected a large amount of genomic data of cultured isolates, okay? So we cultured a lot of these strains in the laboratory, and then we sequenced the genome of these bacteria. And if you have a good reference database of genomes, which very well represents the diversity which is out there, you can take your shotgun metagenomic reads of individual bees and map these reads against this reference database and quantify how much of genome 1 did I have in my environmental sample versus genome 2, 3, 4, and so on, okay? So what we do in this approach is we simply map the shotgun metagenomic reads against the reference database, and then based on the read coverage of these different genomes, we quantify how much, what is the relative abundance of different phylotypes, different sublineages, and different strains in our sample. And you can obviously not only do that for one individual bee, but we did it in one study for more than 50 different bees. And then you can ask the question, how similar are these bees among each other? So this is what Kirsten did. So this was one of her first experimental setups. So she sampled 54 individual bees. They were of 3 different ages. They came from 2 different colonies, and one colony was sampled in 2 consecutive years. So we did the metagenomic sequencing for each of these 54 individual bees. And what is important to note here from this plot here is that 90% of the sequence reads we got, of the non-host sequence read we got, actually mapped to our reference database. So what we can say is that 90% on average, 90% of the diversity we sequence in these individual bees is represented in our reference database, which means that we can very well quantify with the help of our reference database how diversity is structured and distributed in these individual bees we have sampled here. So how did we quantify? So this is really bioinformatics. So I told you before that we mapped these metagenomic reads to our reference database, and then we extracted the coverage, so the read coverage of individual housekeeping genes along the genome position. And since bacteria are replicating, you get a higher coverage here at the origin of replication and lower at the terminus. And to correct for this, we always measured coverage here at the terminus. And you obviously map your reads against all the genomes you have in your reference database. And then you simply sum the coverage over all genomes, which, for example, belong to a given phylotype or to a given sublineage, to estimate and to calculate the relative abundance of that phylotype or sublineage relative to the other community members which are there. And you can do that for, obviously, for every sample individually. So this is very technically, but at the end of the day, what you end up with is a community profile very similar to what you can get with 16S rRNA sequencing. So you have here the relative abundance, you have here your individual bee samples, and you see here the community profiles of these individual bees. And as for the 16S rRNA sequence analysis, you can appreciate that these communities are consisting of relatively few phylotypes. It's exactly the same phylotypes you would identify with the 16S rRNA analysis. There are some differences in these older bees, but this was a relatively small data set to actually test for age. In this case, I wouldn't make any strong statements here about the factor age. However, what you can then do is, since you have not only sequenced the 16S gene, but you have sequenced all the genes, you can actually zoom in resolution and look at how diverse one particular member is. And I take here the example of that Lactobacillus frim5. So in our reference genome database, we know already by doing a genome-wide phylogeny that this Lactobacillus frim5 phenotype actually consists of 4 different sublineages, okay, which are shown here. And we verified that these sublineages are discrete, sequence discrete with our metagenomic data. This is not so important to understand all the details, but the idea here is, since this is based on your reference genome database, you want to make sure that in your actual metagenomes, you don't have additional sequences which are sitting here in between. And instead of 2 lineages, you actually have 1 very diverse lineage, okay? By mapping the metagenomic reads towards your reference catalog and seeing this kind of clearance zone, this gap zone here, you can confirm that you have these sequence discrete populations. So by doing this for all the different community members, we identified that the 8 phylotypes present in the honeybee gut microbiota actually consists of 16 different sublineages or SDPs. Okay, as I told you before, you can also refer to these SDPs as different species. Knowing that they are sequence discrete, you can actually quantify them now, and this is shown here. So for each individual bee, you can now determine how are these 4 different sublineages actually distributed across these 54 bees. And what you can hopefully appreciate from this plot here is that in every individual bee, it looks like all 4 sublineages are present and therefore coexisting. And this is a pattern we observe for almost all the sublineages we have identified. And based on these results, we actually concluded that most of these sublineages seem to coexist and therefore are able to partition the available ecological niches in an individual honeybee. Okay, so instead of just 8 or 10 different phylotypes which are coexisting in a single bee, we can now conclude that there are actually up to 16 different sublineages which can coexist at the same time in an individual bee. However, I told you before that these sublineages can consist of further strain level diversity, right? And you can already see this from this phylogenetic tree here. Although you have these 4 very deep branching sublineages, in each sublineage you have further diversity, okay, which is indicated here by these tip branches. So by mapping reads against the reference database, you cannot discriminate whether you have in your metagenomic sample this strain or that strain, or maybe a 5th strain which is not represented in our reference database. So to quantify this type of diversity, you need to do it in a different way. So we did it in a way that we removed all the genes for the reference database except for 1 per sublineage, and then we mapped the reads against this reduced database, and we quantified the amount of single nucleotide variants in our metagenomic samples, okay? As more of these variants you have in your metagenomic sample, as more strain level diversity you have for a given sublineage. So this is shown here. So on the x-axis, you have these different sublineages or SDPs. So here the 4 for the Lactobacillus frim5, and here for all the other phylotypes. And here you have the fraction of variable sites across all the analyzed housekeeping genes. So this is the total number of variable sites across all 54 analyzed bees. And what you can appreciate, the first thing you should appreciate is that you can have 10% up to 30% variable sites, okay? So there's not too many data sets we can compare this, but there are a couple from the human gut. And in the human gut, using a very similar approach, they actually found that the bacterial species with the most strain level diversity have 3% variable sites. So you can easily appreciate that the strain level diversity in the honeybee gut system is much, much larger than what has been detected in the human gut. Whether this is due to the fact that we maybe have sequenced much, much deeper per species than what they have done in the human gut, or whether indeed the strain level diversity in the human gut is lower than in the bee gut, this needs to be answered in the future. The other point, which is an important take-home message from this plot here, is that when you look at the number or at the amount of strain level diversity per bee, which is shown here as a box plot, it is always lower than the total amount of diversity detected across the 54 bees. So what this means is in a single bee, not all the strains we have detected across our data set can coexist. So strains, while phylotypes and sublineages are coexisting in individual bees, strains seem to segregate into individual bees, okay? So now the question is, which bees actually harbor the same strains, which bee have the same strain profile? And the bottom line is we don't see any clustering according to colony. So the idea that bees from the same colonies harbor the same strains is not fulfilled. This is shown here. So the blue points are bees of colony 2 and the red dots are bees of colony 1. And there's absolutely no clustering or structure in this PCOA plot. So it looks like there's a lot of standing genetic variation, and a bee from colony 1 can harbor very similar strains as a bee from colony 2. We can do a similar analysis in terms of gene content. For example, for this sublineage here, there's not a single pair of bees which harbor exactly the same genes in the microbiota. Okay, in most cases, in this lineage, less than 50% of the variable genes are present in a pair of bees. And many of these variably present genes are actually genes related to carbohydrate transport and metabolism, which suggests that these differences in strain level diversity are of functional importance. So we have followed up on this because we were quite surprised to see no kind of clustering according to colony or age when it comes to strain level diversity. So we therefore questioned that maybe there is an ecological turnover of strain level diversity. I told you before that adult worker bees, they're nurse bees in the first 2 to 3 weeks, and then they turn into foragers. And they not only change completely their behavior, but they're also changing their diet. These bees, they eat a lot of pollen. These bees have stopped eating pollen, and they mainly feed on nectar and honey. So a PhD student in the lab, he has followed up on these findings, and he has sampled nurses and foragers from 16 different colonies at 4 different locations here in Western Switzerland. And to make a long story relatively short, what he found is that on the strain level, he now sees that certain gut microbiota members start to segregate according to whether they're nurses or foragers. So meaning for some sub-lineages, nurses seem to consistently harbor different strains than foragers. For these 2 species here, this seems not to be the case. But for these 2 here, we see clear separation across the principal component one. So there seems to be some kind of ecological turnover of strain composition across colonies. So this is very interesting, and we're currently following up to understand what is really driving these kind of differences. So to summarize this part, what I've shown you so far is that we can measure diversity at different levels using shotgun metagenomics. We can measure diversity at the phylotype level. We can detect and quantify different sublineages, and we can quantify strain level diversity within these sublineages. What we know now is that at the phylotype and sublineage level, we see coexistence. So all the bees across colonies seem to look very similar, and most of the phylotypes and sublineages seem to be able to coexist. However, when we look at the strain level, we see that actually bees start to have individualized gut microbiome profiles. In a single bee, you find different strains than you find in another bee. And this is not necessarily segregating according to colony. So how this diversity, the strain level diversity can be maintained in a colony and also in a single bee, and whether it's functional relevant, this we don't know from these kind of metagenomic analysis. But this is exactly what the bee system makes so interesting, because we can do experiments to address these questions now experimentally, right? So for example, we can ask the question, how is diversity actually maintained in the system? So does diet play a role? Do different bees have a different pollen diet? Are there specific syntrophic interactions between different strains of different species, which allows a certain set of strains to coexist in one bee and another set of strains in another bee? What is the role of neutral processes like priority effects, which means if a given bee is first colonized by strain one, that strain will colonize that bee, while in another bee, another strain arrived first, and therefore that strain will take over the community. So random factors and stochasticity and neutral processes. And what is also the role of bacteriophages, so predator of bacteria. Bacteriophages have been suspected in recent years to play a huge role in shaping and maintaining diversity in microbiomes. So we have different people in the lab who are working on these different aspects. For example, Sylvia, she's doing transfer experiments in vitro cultures, but also in vivo in bees to test which strains are actually able to coexist, and also under which conditions, or different nutrient conditions, or in the absence or presence of phages. I don't have time to go into detail here. The last point, if there's still time, which I would like to touch upon, is what about diversity across different hosts? And for me, one of the most spectacular aspects of the bee microbiome is that when you look across different social bees, people have actually found that the gut microbiome is largely conserved. So different honeybee species, but also bumblebees and stingless bees, they actually harbor very, very similar gut microbial communities, at least on the phylotype level. Okay, and it is believed that this kind of microbiome may have already emerged in the last common ancestor of these social bees. So now the question is, do these bees all share the same strains, or do we find differences in the amount of strains present in different bees? And the hypothesis here is obviously that the ecology of these bees really shapes the genomic diversity we find in the gut microbiome. So honeybees, they build, most of them build very large colonies. Bumblebees, they build very small colonies. They have different social behavior. They have different diets. Stingless bees are extremely diverse. You have more than 500 different species, which have a very different ecology. So although they all harbor the same microbiome, we expect by studying them that we can learn something about how the ecology of the host can actually shape the diversity in gut microbiomes. So the last point I want to show you is one example studies, and this is the analysis, the metagenomic analysis of two very closely related honeybee species, the western and the eastern honeybee, Apis mellifera and Apis cerana. They have diverged about 6 to 8 million years ago. And on the phylotype level, they really have a very, very similar microbiota. So as before, we did shotgun metagenomics of Apis mellifera in Switzerland, but also samples from Japan. And we also sampled Apis cerana from the same places in Japan. We did shotgun metagenomics as before, the same analysis pipeline. And to again, keep a long story very short, is that we found the same phylotypes in both species, but on the level of these sub-lineages, we found 16 as before in Apis mellifera and only 11 in cerana. And most strikingly, only 2 of these sub-lineages were shared among these 2 different host species. So this is shown at the example of Lactobacillus from 5. So for this one, we had identified 4 different sub-lineages in Apis mellifera. In Apis cerana, it turns out that we have a completely different sub-lineages present. And this sub-lineage really dominates across all our samples of Apis cerana. So you can see here that in some of these Apis cerana samples, some of these strains of Apis mellifera specific lineages actually have been able to come in. But what you see here is mostly a very clear pattern in agreement with the host specialization. So you have different lineages and strains present in Apis mellifera than you have in Apis cerana. So although these bees are closely related, they're sympatric, they live in the same places, they have very similar foraging habits, and they seem to have a distinct microbiota, and there seems to be very little exchange of their microbiota. More striking was actually when we looked at the strain level diversity. And what we found here is actually that in a given individual bee of Apis cerana, we can detect much fewer strains of a given species than in Apis mellifera. And as you analyze more bees, you will find more strains, which means as in Apis mellifera, every bee has a slightly different set of strains, but you will never reach the amount of strain level diversity in Apis mellifera. I can skip this. This actually shows the exactly same thing. So we de novo assembled the metagenomes, and the total length of the assembly is much shorter for the Apis cerana microbiome than the Apis mellifera microbiome. So you have much less gene content, coding content in the microbiome of Apis cerana than in Apis mellifera. So much more genomic and strain diversity and gene content in the microbiome of Apis mellifera than Apis cerana. And the question is, why is this? This was to us completely unexpected. We expected to see some differences, but not these kind of very strong differences. And well, there are different ideas now why we see these huge differences. The first one is a concept from macroecology, which is the species-area relationship. As more space a given species has available, or as more space in an ecosystem is available, as more diversity can be harbored in that ecosystem. And Apis mellifera colonies, they're actually much larger than Apis cerana colonies. And that could actually allow much more strains to happily coexist in Apis mellifera than in Apis cerana. So this is one idea. Another hypothesis is that beekeeping has a major influence. So we are doing professional beekeeping on Apis mellifera for many years, and colonies and different host genotypes have been mixed all over the places. And this may also have led to a homogenization of strains of the microbiota, which actually have emerged in different places, okay? And thereby, this has led to all the strains coexisting in the honeybee colonies as we have them today. In Apis cerana, colonies are not being mixed. The beekeeping is much more basic, and we believe that this could also be a reason why we see so many strains actually coexisting in Apis mellifera than in Apis cerana. So these are open questions which we are actively working on. And the question is also, well, since they have more strains and more gene content in their microbiota, is their microbiota more beneficial or less beneficial than the microbiome of Apis cerana? And we have a couple of new projects lined up. One is to look at more different species of honeybees. There's not only Apis cerana and Apis mellifera, but there's also Apis dorsata, Apis laurea, and Apis adreniformis. And together with the collaborators in Malaysia and Kenya, we plan to do a much bigger sampling of all these bees and carry out metagenomic analysis. We have a similar project also on stingless bees. So the key points of my talk of today, the take-home messages would be that bees harbor highly specialized gut microbial communities. The microbiota impacts bee health, and there's really a good potential and hope for future applications based on the microbiome, or what we learn from the microbiome, or methods we can establish based on microbiomes. The microbiota of bees is tractable experimentally, which makes it a very good and versatile model to ask a lot of different questions about gut microbiomes. And there's a high level of genomic and strain level diversity. And the question is, what really drives diversity in microbiomes, and how does diversity impact actually the health of an organism, in this case, the bee? And I think, again, the bee model is perfectly well suited to address these questions, both by looking in natural populations of bees, but also by carrying out experiments in this honeybee model. We see differences in the microbiota across related bee species, so there is clear evidence for host specificity. Different bees carry different strains of the same phylotypes. And also here, the question is, what kind of evolutionary mechanism has driven this kind of divergence in the microbiota across these different bees. So with this, I'm at the end of my talk. I would like to acknowledge many different people who have been involved in this research. So this is our current group in the lab. So I could only touch upon a couple of topics which we are currently following in the group. A couple of collaborators, Uwe Sauer, Ria Miyazaki, were essential, were very important for the research I've shown you. I also want to point out that we have a couple of projects also on the cheese microbiota. Swiss, cheese is very big in Switzerland. So we have a project with the Agriculture Research Station on cheese starter cultures. And we are collaborating with a couple of people at our hospital on the human microbiome, especially the lung microbiota. And obviously I shouldn't forget to mention all our different funding sources. The NCCR microbiome is a huge consortium in Switzerland. Many different research groups are involved in this, and we have a common interest in understanding, controlling and manipulating microbiomes for better health. So with this, I thank you for your attention, and I'm happy to take some questions.
Thank you for that very illuminating talk that took us right from the basics of microbial science in bees to open questions in the field today. I will be moderating the Q&A now. So if you're on YouTube, please feel free to put questions in the YouTube box, comments box. And if you're on Zoom, you're welcome to unmute yourself and ask your question, or to put a question in the chat and I can ask it. So we've had a few questions. One is, if there were only 1,000 or around 1,000 ions that were found, how did this translate to over 3,000 metabolites?
Well, there were 17,000 ions detected, I think, but you can only annotate a small fraction of those, okay? The problem with untargeted metabolites, at least the method we were using, is that a certain ion can have 3 different annotations, okay? So given mass, which you measure, can be either annotated as compound 1, compound 2, or compound 3. So in this case, you're not completely sure which of these 3 compounds you actually detected in the gut. So this is one of the drawbacks of these methods. But we focused on compound groups which either had a unique annotation, so one ion could be clearly assigned to one metabolite, or we focused on metabolites which were not only represented by one ion, but by many different ions. For example, these flavonoids were represented by many different ions, and even though one ion had several annotations, another ion had only one annotation, and a third ion had maybe 2 annotations. So it's true that this is a drawback of the method, but well, that explains why you can have 3,000 metabolites detected by 1,000 ions, potential metabolites, okay?
Thank you. We have another question from Zoom about holobionts. So they say that, holobionts is obviously a controversial theory, but when you see the deep evolutionary conservation of these strains with the honeybees, is there sort of more support for holobionts in the bee systems then?
Well, that's difficult to say at that point. I think what is very challenging to address is if there's like co-evolution in a strict sense, like, does the host affect the evolution of the microbiota, which is maybe easier to answer, but vice versa, does the microbiota actually influences the evolution of the host? And I think to really think in terms of holobiont, that things, you know, evolve as a holobiont, you need to show that the microbiota also has influenced the evolution of the host, which I find quite difficult to prove. But it's a very controversial topic, I agree.
Thank you. Okay, so we have someone on Zoom who would like to ask their question themselves. So they're going to ask now.
Yes,
yes.
Sure.
Thank you for the talk. It was very interesting. So my first question is, at the start, you were saying that to control the species of the bacteria, you gave one species at a time for germ-free bees. I was wondering why you didn't give everything except for one species. To make sure that, for example, if there's interactions between different species to make one thing, then that cannot be detected if you only give one species at a time.
Yeah, that's a great point. I think that would be a highly complementary approach to the one we took. Obviously, if one member is missing, another member may take over that niche, okay? And then you may not be able to detect any metabolite, any relevant metabolite change. On the other hand, if you only colonize with one member, that member may take over niches of other bacteria which are not there. So I think both approaches have pros and cons. Ideally, you know, you would test them both. One problem indeed is that if you do monocolonizations, the bacteria may behave completely different than when it is in the community, right? So that would speak in favor of the approach you're suggesting. So that's a great suggestion, and we have discussed this also forth and back in our lab. If that would be a more if that would be a good complementary approach to use in the future.
Okay, thank you. And my second question is, you were saying that the diversity within the colony of the bacteria in the guts are partly due to like what they do. So what do you think might be other factors that determine the diversity within a colony, as in like is there much individualism within bees?
Yeah, I mean, this is right to our core question. I mean, right now or in general. So one thing so bees obviously they collect, so they eat nectar and pollen, right? But pollen is not equal pollen. I mean, pollen can come from many different plants. The structure of the pollen is different. The nutritional value of different pollens may be different. So you could think that depending on what kind of pollen and how much of one pollen one bee ate, you know, one strain is better off than another strain, okay, and will win the race. But it could also just be neutral processes, right? So, you know, these bees, they emerge, they become adults, and then they're exposed to the hive environment and to thousands, hundreds of different bees. I mean, you see it in my background, right? So imagine you come out in a brood cell right here, you will immediately be in contact with 5, 6, 10 other bees, and they may harbor a given set of strains, and these are the strains you will get, but you will not get the strains of the bees which are sitting up here, right? But the bee which is emerging there will get these strains. So maybe just a random factor that the strain which arrives first in the place will have a huge advantage over a strain which arrives later. And thereby you can get this kind of segregation of strains into different individuals. So that would assume that all the strains are the same, you know, they're genetically slightly different, they have different genes, but in their function that they're exactly the same, they're neutral, okay? and it just matters who arrives first at a given place. So this is called priority effects. It's the same with like island geography. So you have, you know, empty islands, and then, you know, you arrive as a bird, for example, and the one which arrives first just has filled already the niche, and the next bird which could do the same thing just can't. So, well, there are different aspects. I mean, diet, if you believe, so Apis cerana seems to have less strain level diversity than Apis mellifera, okay? But based on what is known from the literature, they have a very similar diet, and they forage on the same flowers. So you would expect that the nutritional content in their gut is the same. So why does one bee have fewer strains than another bee if diet would be the factor which matters? So these are the questions we are looking at, or we think about how we could look at.
Okay.
Very good question. Thanks.
Thank you for those questions. I have another question off Zoom. Thank you very much for the great talk. You mentioned that A. mellifera and A. carina are closely related yet harbour differing levels of bacterial diversity. I was wondering how closely you have looked at the genomic differences between A. mellifera and A. cerana, whether there could still be genetic specificity in the host that facilitates recognition specificity for bacterial species.
Yeah, I mean, that is possible. I mean, we know really nothing about to what extent the host controls or regulates which strains or which species can colonize its body, right? It could be that there's some type of filtering going on via the immune system or some other molecular mechanisms which allow the host to just select certain strains or to select more strains than another species. I'm not an expert on honeybee genomics, and I'm not sure how much you could learn from looking at their genome. But it's completely possible, right, that the host actually plays a huge role in what kind of strains can actually colonize. So what we have done, and others a little bit, is that we have generated germ-free honeybees, for example, and then we try to colonize them with strains from bumblebees. Okay, so bumblebees is the same as with Apis cerana. They harbor the same kind of phylotypes, but different lineages of that phylotype. And you can colonize a honeybee with a bumblebee strain. It looks like the colonization levels of most of these bumblebee strains will be a little bit lower in honeybees than the honeybee in their native host. But they still can colonize, okay? So the host seems not to have, in that case, a strong, does not play a strong role in regulating who can colonize and who cannot colonize. But what we have observed is as soon as you put that non-native strain in competition with a native strain, that non-native strain has absolutely no chance to colonize that host anymore. So it's outcompeted by the native strain. So the bottom line is no, we don't know. It could be possible, we don't know what's the role of the host, to what extent the host can control.
Thank you. Are there any other questions either on Zoom or on YouTube? Okay, I think in that case, we're going to wrap up. So thank you very much for this talk. It's been very interesting and very informative. And it's just been especially nice to go from the basic setups to the current questions in the field about diversity, and also see the important applications this research may have. So thank you for the talk, and thank you to our listeners for coming, and good night.
Yeah, thanks a lot. Thanks for the great questions. That was really good. Bye-bye.
Is live streaming
Yep.